Hi, Michelle Glass here, and we are continuing our discussion of organic macromolecules or biomolecules for our second half of chapter 2. So we have already talked about carbohydrates and lipids. Now we're ready for proteins, and then we have one more video presentation on nucleic acids. So make sure you're checking that one out as well. So we are here for our discussion of proteins, which are super cool molecules. Essentially, you're just one big sack of proteins. When we talk about proteins, we will see that the list of their functions is long. And we will also see that the DNA molecule, your hereditary molecule, is responsible for telling your cells how to make those proteins. So as we think about traits that are inherited or passed on through the family, all we're passing on really is that recipe for how to make those proteins. And so all of those things that we think about as hereditary traits, all at their core are just based on our proteins. So that is why they are so cool. Okay, so let us get started. To begin, we have a whole list of functions to talk about for our proteins. Remember, the production of those proteins is determined by your DNA molecule, your hereditary molecule. Our proteins are going to be responsible for catalyzing all chemical reactions in the cell and in the body, and we call that group of proteins enzymes. We'll talk a little bit more about what this means toward the end of this video, but essentially, you have to have an enzyme present in order to have a chemical reaction happening. So we can control what chemical reactions a cell can do based on what enzymes are available. We also see that proteins are important in communication, just like we had lipids. Forming some of our hormones. Some of our hormones are made from proteins. We will also see proteins forming cell membrane channels and receptors on those membranes. So this is really important in how cells communicate with each other, how we get things in and out of the cell, how we get things to, again, bind and communicate with the cell. So that's going to be another big job. The structure of our body at every level can be related to those proteins. So when we're looking at the cell structure, there's going to be proteins involved in the formation of what we call cytoskeleton. When we look at our tissues, you've already seen some of these proteins. When you were looking at your connective tissues, you saw collagen and elastic and reticular fibers. Those were proteins. When you look at your muscle tissue, when you look at those striations for skeletal muscle, you are actually able to see them through the microscope, some of those contractile proteins that are involved there in all muscle contraction. So that takes us to that next one. And then also our proteins are important in our immune response. So we have both these little name tags hanging off of the outside of our membrane that lets our body know which cells belong to us and which cells don't. And then in addition to that, you've heard before of antibodies, and those are actually protein molecules. So a big part of our immune response is going to be connected into our proteins. As we talk about proteins, we have to spend some time talking about the structure of the protein, because the structure of the protein has a direct relationship to the function of the protein. For proteins to work, they basically have to recognize and bind to other molecules. And so the shape then of the protein determines its ability to recognize and bind. So that's meaning that it's determining what kind of function it can do. The structure of your protein, we've already mentioned, is coded for in that DNA molecule. So remember, the DNA is like the recipe for how to make the protein, so that means its shape. And then we will also see that the environment of your cell and the body fluid will also affect protein structure. So temperature and electrolytes and pH can all have an effect on protein structure, which has a direct relationship to its function. So one of the reasons that these characteristics are homeostatically regulated is to ensure that our proteins are shaped the right way so that they can function the right way. If you in any way alter the shape of your protein, you alter its function. Now, sometimes you can just tweak it a little bit and it just, doesn't work quite as well as it should. Sometimes you change the shape to such an extent that it doesn't function at all. Sometimes that change of shape is permanent. We call that being denatured. Sometimes that change of shape can go back and forth between like the correct form and the incorrect form. When we talk about protein shape, it is a complicated shape. And of course, that makes sense because the shape has to be specific for it to recognize and bind to the molecules in order to function correctly in our bodies. So we have actually 4 levels of protein structure that we need to talk about. We have a primary, secondary, tertiary, and quaternary, which you can abbreviate each of those as the number and then put the degree symbol at the top. So here we're looking at our description of the primary structure of a protein. The monomer for a protein is an amino acid. So our primary structure is a chain of those amino acids. An amino acid is going to have a carbon at the center and then 4 distinct things binding to that central carbon. So we're going to have an amino group. So that's going to be nitrogen with hydrogen. We have a plain old hydrogen, and then we have a carboxyl group. So the amino group gives us the amino part of the name, and then the carboxylic acid part gives us the acidic part of the name. And then we have this generic R. Remember, we saw this in our discussion of functional groups. The R here is analogous to that X in algebra, where it can stand for any number. Here, the R can stand for like a range of different groups here. There are 20 different amino acids, and that means there are 20 different R groups. So all of the proteins that are produced in cells are combinations of these 20 amino acids. So here is a nice image from Google. We are not memorizing this, thank goodness, right? But what we do want to see is pay attention to those R groups, see how much variety there is there. Some are acidic, some are basic, some have charges, some are polar, some are non-polar. So this will matter when we start looking at especially that tertiary, that 3rd level of structure for our protein. So we'll see interactions between these R groups will affect the shape at the tertiary level for our protein. Okay, so we said that our primary structure is a chain of amino acids. We will describe those amino acids as being connected by what's called a peptide bond. So we can see a picture here from our textbook showing us, again, here are 2 different amino acids. We're going to remove the water, performing a dehydration synthesis reaction in order to form our peptide bond. Or if we're eating proteins and breaking them down into our monomers, then we're going to do a hydrolysis reaction in order to get those building blocks separate so we can make whatever thing our cell wants to make, not the thing that we ate, okay? So we're seeing dehydration synthesis and hydrolysis reactions in the formation of each of these macromolecules, going from monomers to polymers, from polymers back to monomers. You can describe this chain of amino acids linked by a peptide bond as a polypeptide, where the prefix poly means many. So polypeptides are like a small version of a protein. It's talking about a chain of amino acids without a whole lot of that folding necessarily associated. The secondary level of, let me go back. Let me show you my visual aid. Forgot. We're going to do this in class 2, so you'll have opportunity to actually perform it. But I'm going to use here a pipe cleaner with beads to represent my primary structure. So I don't have a zillion beads, I just put a few on here, but each of those beads, let's just pretend is an amino acid. So here we're just looking at our primary structure of our protein, chain of amino acids linked together by peptide bonds. Okay, now we're ready to look at that secondary structure. And for the secondary structure, we're going to see that there will be hydrogen bonding. Remember, hydrogen bonding is interactions within a molecule. And we're going to see interactions occurring between, along the length of this molecule. And there can be two different ways that we will see secondary levels. So two different types of folding. So I've already done one for you here. This is the alpha helix. So it just looks like a spring. And then I'm going to show you the beta pleated sheet with my pink strand. So you're just folding it up like this. Remember, with hydrogen bonding, you're basically looking at, attraction between opposite charges and repulsion then between like charges. So the things that are attracted are going to fold up to be as close to each other as possible. Things that don't like each other are going to try to get as far away from each other as possible. So secondary folding is automatically happening based on interactions along the strand. Hydrogen bonding, it can come in the form of either a beta pleated sheet or alpha helix, just depending on what those interactions are. Okay, now we're ready to talk about that tertiary level. The tertiary level is dependent on the environment. So this is where we see hydrogen bonding between those R groups and the aqueous solution. Remember, aqueous means water-based, and we're talking about proteins that are present inside of a cell or in the fluid surrounding the cell, all of which is an aqueous solution or watery-based solution. Water, remember, is polar and a universal solvent. So you're going to see lots of other polar molecules and lots of ions present in the fluid inside and the fluid surrounding the cell. And so you're going to see then that these R groups, if they're polar or nonpolar, are going to have different reactions with that environment. So again, this is really one of the key reasons why we have to homeostatically regulate our internal body so that our tertiary structure of our protein will be shaped correctly. So let me show you how I like to represent that. This is super high tech. So I have my alpha helix, and then I'm just going to represent here my environmental, my tertiary level of folding. And let me do that here as well. Okay. And then, whoops. Then we're going to look at our quaternary level, which is described as a complex. Here we can see the globular version would be like if we just bring these two subunits together. That's my quaternary structure. If I want to represent fibrous, then I might show it like this. So fibrous is more like those collagen fibers and those reticular fibers and all of those that you studied as part of your connective tissues especially would be that type. Again, this structure is significant because it determines the functionality of those proteins. Here's a nice picture from our textbook showing us all of these different steps. So we have our primary structure chain of amino acids determined by our DNA molecule. Here's our secondary level, can either be the alpha helix or the beta pleated sheet. So here we see our hydrogen bonding along the length of that strand. Here is our tertiary structure. Remember, that's altered by our environment. And then here's those complexes, so either the globular or the fibrous. Remember, all of this has to come out exactly just so in order for that protein to work exactly just right. Any small alteration will alter its function. Sometimes that means it's just not as good as it could be. Sometimes it means it's not working at all. When we're talking about our proteins, we've mentioned enzymes specifically as a special group, so we do want to make sure that we're familiar with that. And then I talked about those name tags on your cell membranes, and we can talk about that group of proteins as glycoproteins. So the glyco there means that it's got a carbohydrate component. To finish up, let's talk a little bit more about those enzymes, which can be described as biological catalysts. Catalysts are chemicals that are involved in the chemical reaction but not changed in any way. So we typically draw them above the arrow of our equation. And essentially, they are speeding up the rate of our reaction so that it doesn't take years to happen, but it does happen safely in the cell as well. These enzymes are going to determine a cell's metabolism. So in order for a chemical reaction to occur, you have to have a specific enzyme there for that reaction. Remember, metabolism is both anabolic and catabolic reactions. The cell can control what chemical reactions it does by controlling which enzymes are being produced and also which enzymes are active. So you have to sort of sometimes turn on enzymes, or you can turn off enzymes to stop a reaction from happening. Your enzymes are very specific, so we often describe this as like a lock and key model. Enzymes match up with specific molecules called substrate, and each enzyme just matches up with a particular substrate, just like a single key is just fitting into the lock. Essentially, when that substrate binds in with the enzyme, it just makes that reaction happen easier. So it could either be anabolic or it could be a catabolic reaction. Reactions are dependent on that enzyme having its optimal shape. Remember, the shape determines its function. So it is affected by both temperature and pH. In order for enzymatic reactions to happen well, the enzyme has to be at its optimal shape. We can see if those conditions are too outside of the optimal conditions, it can affect your enzyme shape. Especially heat is one that we know will denature your enzymes. That's usually a permanent denaturing, meaning that it's changing the shape. You know this. You cook food and it changes texture and taste and color sometimes. And that's all linked into the denaturing of those proteins that's part of that food. And you also know you can't take cooked food and turn it back into raw food. pH can also then denature enzymes if it's too acidic or too basic, if it's outside of whatever is optimal for that particular enzyme. There are times when these conditions denature proteins, which it's not permanent, so sometimes proteins can denature and then renature, but sometimes it is a permanent shape change. And that is it. So stay tuned for the last video.